Good day. I am Brian Hoffland, President of Research to Prevent Blindness, and I want to welcome you to RPB's Lunch and Learn, Eye on Stargardt Disease. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, and eradication of all diseases that destroy sight. RPB was founded in 1960, and over our 64-year history, we have awarded more than $400 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. We're incredibly proud of the breakthroughs made possible by RPB-supported researchers, but there's still so many treatments that need to be developed, so our work continues to fund the breakthroughs of tomorrow. In today's educational session, you will hear from two RPB-supported talented scientists about Stargardt disease and its causes, impact, and cutting-edge research. One of our speakers, Dr. Jason Commander, received an RPB Career Development Award, and the other, Dr. David Gam, received an RPB Nelson Trust Award for retinitis pigmentosa and an RPB Catalyst Award for stem cell research in AMD. Thanks to all the generous supporters of RPB who make our work possible. I'd especially like to thank Alkeus Pharmaceuticals for their support of today's educational session. The importance of supporting medical research has never been so apparent. We thank you for your partnership to support innovative vision research and to ensure that new breakthroughs are possible. If you have not yet supported RPB, please consider doing so to help make sure that we are able to continue these breakthroughs. Getting back to today's session, let me introduce our moderator, Seemi Khan, MD, MPH, MBA. Dr. Khan is an internist and nephrologist with expertise in rare disease, immunology, and rheumatology. She joined Alkeus in January as Chief Medical Officer. She has a track record of bringing rare disease treatments to patients in need across multiple therapeutic areas. Prior to Alkeus, Dr. Khan served as the Chief Medical Officer at Reata Pharmaceuticals through its acquisition by Biogen, where she oversaw all clinical programs from early stage to commercialization. Dr. Khan's experience also includes senior leadership roles at Mitsubishi Tanabe Pharma America, Cork Pharmaceuticals, Abbott, and AbbVie. As moderator, Dr. Khan will introduce our two speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes and I will serve as timekeeper. I will give each a warning at the 18-minute mark. We want to make sure that we leave plenty of time at the end for robust Q&A session. And with that, I'll turn it over to you. Seemi, please take it away.
Thank you so much. Thank you, Brian. Really appreciate that you are arranging this event today. So today our event is mainly about the Stargardt disease, a most commonly inherited retinal condition that leads to progressive vision loss and often beginning in the childhood or adolescence. Understanding this disease is very crucial to both patients and their families, as it helps to them to anticipate the challenges ahead and explore potential treatments. Stargardt disease impacts daily life in profound ways, but staying informed about the latest research and management options, patients can improve the quality of life across clinical trials and have an access to them and find the right support system. This webinar is a key step in educating and empowering both patients and the researchers focused on future therapies. We at Alkeus are deeply committed to developing innovative therapies for retinal diseases. Our commitment is evident in the advancement of gildoretinol acetate, a novel oral investigational treatment designed to slow or halt the progression and preserve vision in this therapeutic area. With that, now I'm very pleased to introduce our first speaker, Dr. Jason Commander. Dr. Jason Commander is an assistant professor of ophthalmology at Harvard Medical School and a member of Massachusetts Eye and Ear Inherited Retinal Disorder Services and Retina Service. He specializes in the management of inherited retinal diseases and is deeply involved in genetic research as part of Ocular Genomics Institute and Birmingham Laboratory for Retinal Degeneration. Dr. Commander is also a leading investigator and surgeon in several clinical trials, searching new treatment for these diseases. Today, he will be presenting inherited retinal disease with focus on Stargardt disease epidemiology, such as both prevalence, incidence, current management practices, in addition to the current landscape of clinical trial. With that, Dr. Commander.
Thank you so much, Dr. Khan, for that nice introduction and for the opportunity to be here today and share this with you. Let me share my screen here. So I'd like to start with an overview of Stargardt disease. And the first thing that people always ask is, what does this word mean, Stargardt? And what does it have to do with stars or what? So actually, it was like many of the old diseases in medicine, it was named after a doctor. So good old Dr. Carl Stargardt published the first description of it in 1902, so over 120 years ago now, and it was in German. But if you translate the title there, it's something like on familial progressive degeneration in the macular region of the eyes. And he had some pictures in the back of this article. Actually, they're not pictures, they're drawings or paintings, because they didn't have a fundus camera back then. So you see the name of the artist at the bottom of the screen there. But for over 100 years old, these are really nice, pretty accurate. They didn't have the kind of fancy cameras that we have now. Hopefully you can see my mouse. And for those of you who may have trouble seeing it anyways, I'll be narrating everything so you won't miss out. So the white is the optic nerve and the blood vessels are coming out. And on the left, it's giving an example of kind of earlier severity of Stargardt, where there's some pigment changes in the center of the retina. They showed some little striations. These little dots, in reality, this is the one part of the drawing that's not good. They're usually like little elongated lines that we call flecks. And they reminded somebody of like a shape of a fish's tail, so they call them pisiform flecks. And so if you hear that jargon, they're just describing the deposits that happen in the retina. And on the right-hand side, there's a drawing of a patient with more advanced Stargardt disease. So there's this area in the center of the vision and the center of the macula where it's worn out, and the vision's not going to be good there, but the vision all around it can be okay. So what is it? Stargardt disease is an inherited disease of the retina that causes these macular problems. And again, the first thing when a lot of patients say, when you say, well, it's an inherited disease, they say, well, my mom's vision is great, my dad's vision is great, nobody in my family has this. And it's because Stargardt disease is recessive, so you can be a carrier without any disease, and then it's just the unfortunate luck of getting one dose from one parent and one dose from the other parent, who both have normal vision, normal eyes, and if those two bad copies of the gene come together in one person, then you're the one that's affected. And almost all of the cases of quote Stargardt have this recessive inheritance. So it shows up in brothers and sisters mostly, and it's only 1 out of 4 statistics wise. And so with the kind of family sizes that we usually have in this country, it's usually only one person. Okay, so terminology. Now I realize that the terminology isn't medically important, and we're going to get to the clinical trials and what we can do about it and all that more important stuff. But again, a lot of people get concerned that one doctor said one thing and one doctor said another thing, or do I have it or do I not have it? And so I'll tell you the way that I think about it. And, you know, knowing that different doctors may describe it differently. So in the old days, people with macular problems were divided up into juvenile macular degeneration and senile macular degeneration. And we don't say senile anymore. They've changed it to age-related. So age-related macular degeneration is a very common disorder of the retina, where people that are older, say 60s, 70s, 80s, 90s, can get a wearing out of the central retina, the macula. And it's not, and that one is genetic, but not strongly genetic. So it may happen to some people in the family, but not others, to some brothers and sisters, but not others. And the kids, you know, back in the day, the kids described with juvenile macular degeneration, most of them had Stargardt. And with the genetic revolution that happened, now when we do genetic testing for people, we can kind of like subcategorize those juvenile macular degenerations more carefully. So the gene, the name of the gene that causes Stargardt disease, the acronym for it is ABCA4. It doesn't matter what it stands for. It's a vitamin A transporter in the retina. And so now, instead of saying juvenile macular degeneration and senile macular degeneration, now it's macular dystrophy, which kind of implies that it's inherited, and age-related macular degeneration, or AMD. So most macular dystrophies are Stargardt disease, and most of the people with Stargardt disease kind of present in childhood or early life. But we do have people that don't know that they have it until they're older. 60s, 70s, we have some people come in who they thought they had age-related macular degeneration, but because of the way the retina looked, we did genetic testing and we found that it was actually Stargardt disease. All right, so where and how does it affect the eye? So we talked about it a little bit already. So the center part, the central 10 degrees or so of the vision corresponds to the macula. That's just the center of the retina. And it causes a very slow wearing out of that region that affects the central vision. So who is affected? So it's rare. It's 1 in 10,000 people. But you wouldn't know that here because we have people coming from all over to get their Stargardt disease evaluated, et cetera. And if you're one of those 1 in 10,000 people, then it's quite important for you. And there's a lot going on for a rare disease, which is part of why we're talking today. What are the symptoms? So with mild Stargardt disease, there can be no symptoms. So sometimes we get kids in here who have an older sibling that has it, or that was just discovered routine on an eye exam. Somebody saw some of these flecks that I was talking about, and they may not have any symptoms at all. But often in a mild case, you can have distortion or decrease in the central vision, the reading vision. Typically, the side vision, the peripheral vision, the night vision, the color vision is all good. It's affecting the central vision. When it's more advanced, like more and more of the central retina can be affected such that reading vision is affected. So how it tends to affect people in real life is they could have difficulty reading or difficulty driving at night or difficulty driving. This is in contrast to some other inherited retinal diseases, like retinitis pigmentosa is the other kind of like most common, most famous one, where people lose their side vision first and keep their central vision for the longest. So Stargardt's is the opposite, where the center vision is more at risk, and the vast majority of people keep all of their side vision their whole life. Current management options. Well, we really don't have anything, you know, which is why the whole rest of the talk is talking about what's coming up. So we tell our patients, wear sunglasses outdoors. We don't really know if that makes a difference or not. I'm not too strict about it. Don't take vitamin A supplements. If you're, we can talk more about that later, but that's really all we have in vision rehab, et cetera. So it's actually quite an exciting time for potential treatment strategies for Stargardt disease. And I've divided them up into like for milder or intermediate disease and for more severe disease. Dr. Gam after me is going to be tackling the regenerative medicine part of this. So for milder disease, we'll get to that in a minute. I'll also add in, it's not really a treatment strategy, but prevention. This is sponsored by Research for the Prevention of Blindness and Vision Rehab, which I'll throw in. So gene therapy. So the fundamental problem in Stargardt disease is that this ABCA4 gene is not working. So it's either not working at all or it's working not enough. And so one strategy is to deliver a correct copy of that gene into the cells in the retina. And this was done about 10, 12 years ago with a DNA carrier that's a modified form of a virus that has like the virus parts removed and you just have the DNA that you want in it. The Stargardt gene is very big and it doesn't fit in the preferred cargo carrier. So they picked a different cargo carrier called lentivirus, and Sanofi sponsored that trial, and it didn't work well enough to go on. And then the field sat for like 10 years. In the meantime, Luxturna, which was the first FDA-approved gene therapy for any inherited disease, was approved for a rare inherited retinal disease, not Stargardt disease, but for the gene that one treats is called RPE65, is the acronym for that one. And that causes Leber congenital amaurosis or retinitis pigmentosa. I love treating people with that. It's really cool. I'll show you a surgery video later. So now, for the first time, we have the new Stargardt gene therapy trial starting up. And the way that these are different is that because the whole instructions for the gene don't fit into the one version of the virus, it's really good at delivering the gene to the retinal cells called the AAV. People have figured out how to make use of the cargo capacity that's there. So one company, Splice Bio, has figured out a way to deliver two halves and have them come together in the cell at some efficiency, which is really neat. And Avant-Garde Bio is doing a similar dual vector approach. A slightly different, but almost a slightly different approach was taken by the company called Acidian, and they are inserting a piece of DNA that makes RNA, and then the RNA kind of finds the broken copies of the ABCA4 RNA, kind of fixes the first half of it. So this one is designed for people with their DNA mutations in the first half of the gene. Ocugen is also doing a gene therapy study. This is kind of a modifier gene approach where they're not putting the ABCA4 back in, but they're delivering another gene that's supposed to help the health of the retina. That one's the Guardian trial, and it was being done in Arizona and Texas. I think the phase 1 is complete. I'm not sure if it's in phase 2 right now. Another approach, which a lot of people ask about, and you may hear about for other diseases, is gene editing, where you actually go in and fix the mistake in the gene. This, to my knowledge, hasn't made it to humans yet in Stargardt disease because there's so many different changes in the DNA, so many different mutations that can cause Stargardt disease that you wouldn't want to have to make a different drug for each one. And none of them are like so super common that that there's tons of people to do a specific product for. So it's nice that all the other ones on the slide here you can do for, no matter where the mistake in the gene is. So how does this work? So it really is a little bit like magic. So you take the DNA sequence, either of the two halves of the gene or of the RNA piece that you want, or whatever the strategy is. You grow up you put that DNA into bacteria. You grow up that bacteria in the laboratory. You purify that bacteria. You put it in different cells to make that, to package into this virus that's had the virus parts removed. Then you purify that and it becomes this clear liquid. And I get this tube of clear liquid in the operating room, and my job is to inject it underneath the retina. And then after that the viruses in that clear liquid are very good at injecting the DNA into the retinal cells. The DNA gets into the retinal cells, and then whatever it is that's supposed to be produced is. So for the simplest case with the Splice Bio or Avant-Garde, where you put the two halves in, the two halves get in there, they produce the enzyme or the molecular machine that's coded by this ABCA4 gene, and then it can better process the vitamin A in the cell. So here's a schematic of what that looks like. So this is a vitrectomy, where you take the vitreous out of the middle of the eye. The little instrument there is called a vitrector, and it's trying to show that removing the gel from the eye. You don't really need the gel in the eye except for the prevention of cataract. This is all like microsurgery. And then we take a tiny little tube, the width of a human hair, and we inject about 3 drops of liquid underneath the retina. And you can see it's a simulation of the retina kind of bubbling up underneath it. But I'm going to show you a real one in a minute. So this is on a cellular level, and that's one of the things that's cool about this, is that this is really working on the cells. Here's a real surgical video, and it doesn't have anything gory. I cut out the eye picture at the beginning. Some people don't like that. So this is the vitrectomy part, and then we're removing some strands of the vitreous, preparing the site for the injection. And that's a tiny little tube going up there, and then you'll see the liquid going in and going across the fovea, across the macula, and kind of filling up the central parts of the retina. And so there's, you know, hundreds of billions of little virus particles in that clear liquid, and they're busily injecting that DNA into the retinal cells to try to fix this at a molecular level or treat it at a molecular level. Then for Luxturna, we do it in both eyes. So like a week later, we was doing in the other eye. There is some of the liquid is going towards the edge there, which happens sometimes. And so amazingly, the liquid soaks in over the next day or the next week or so. And when we come back, it looks like nothing's happened. But really, everything that's happening is taking place on a molecular level. And Luxturna was approved because it causes an increase in night vision in patients. This is a totally different disease than Stargardt disease, and that's not expected to happen with these kinds of gene therapies. The gene therapies are kind of targeted to try to slow down the progression of the disease. That's gene therapy. You can ask me more about it during the question period. How are we doing on time? I didn't start my timer at the right part.
Jason, you're at the 17-minute mark.
Oh, okay, good. So there's a number of strategies using pills or oral medications instead of subretinal injections. And Dr. Khan could tell us all about this. The Alkeus is working on medicine called gilded retinol, which is supposed to improve the chemistry of vitamin A metabolism. And they have done or are doing 4 clinical trials for this. This medicine potentially is the one that's potentially closest to getting approved, because TEAS 1 is done. TEAS 2, the results are coming out at the beginning of next year, I believe, and TEAS 3 and 4 we can get into that if we have time. There are some other strategies, including a molecule to slow down the chemistry in the retina, or metformin, which is being done at the National Institutes of Health. There's an approved molecule for age-related macular degeneration that gets injected in the eye. Iveric did a study of this in Stargardt patients, but they never released the data, which usually isn't a good sign, but I'd love to see it. For the final slide, I just wanted to throw in something if you're, for those people in the audience who have Stargardt, if you are planning a family and going to have children, one option that you have with modern medicine is to have your partner screen to see whether they're a carrier for Stargardt disease. If they are, then it's not everybody has an access or preference to it, but you can do IVF with pre-implantation genetic diagnosis to have a kid that doesn't have it. It's about like a 1% chance that that someone could have it. So it's low, and if you don't have access to that, then you shouldn't have any guilt. But on the other hand, 1% is like the same as lots of other things that they screen for in obstetrics to try not to try to detect. I'll put in a plug for Vision Rehab while medicine is working on all these fancy things like learn all about technology. That's really like more than Braille say how people are getting things done these days who have limited central vision due to Stargardt disease. And I'm happy to answer questions about that. So thanks for this opportunity.
Thank you, Dr. Commander. That was a very informative talk. Now I'm pleased to introduce our second speaker, Dr. David Gamm. Dr. Gamm is a professor of ophthalmology and visual sciences at the University of Wisconsin School of Medicine and Public Health, and the director of University of Wisconsin School of Medicine and Public Health and McPherson Eye Research Institute. He's a pediatric ophthalmologist and adult strabismus specialist. He's leading cutting-edge research in cell-based therapies to treat retinal degenerative diseases. Dr. Gamm contributes to the stem cell research extend across several prestigious programs, including the Weizmann Center Stem Cell Research Program and the University of Wisconsin Stem Cell and Regenerative Medicine Center. He will be presenting today the concept of regenerative medicine and progress in the ongoing stem cell research. With that, I'm just passing on to you, Dr. Gamm.
Thank you very much for the kind introduction, Dr. Khan, and also to Dr. Commander for leading off and giving such a great introduction to Stargardt disease and approaches to early to mid stages of the disease. Where I take over here is kind of talking more about later stages of Stargardt disease, so more advanced stages. And why is it that we think differently when we think about patients who have had Stargardt for a long time or are at their end stages of the disease than we do folks who are in their earlier stages? And to talk about that, I want to just do talk a little bit more about just basic Stargardt disease. And so here's a normal retina. When ophthalmologists look through your dilated pupil to look at your retina, this is what we hope we see. This is a nice healthy retina, and the center portion is right here. It looks a little more red, and that's the center of your macula or the fovea, and that does all of the high acuity seeing that we do, what we use to recognize faces and to read newspapers. So it's the most important part of our retina. And if we now peel that retina out, which looks just like a piece of thick cellophane, and look at it on side, this is a cartoon, of course, we see that it's not just a piece of cellophane. It's actually a complex, very elegant layer cake of different types of cells. Deepest within the retina are two cell types called the RPE cells and the photoreceptors. Photoreceptors are rods and cones. Cones detect color, light, high acuity vision. Rods are more peripheral vision and night vision. And they are pound for pound the most metabolically active cells in your entire body, and they're designed to detect light. So they have structures called outer segments that are deepest within the neuroretina itself. And those outer segments intercalate with processes from the retinal pigment epithelium. And the photoreceptors and the RPE together comprise the outer retina. And they're like Batman and Robin. All of the credit goes to the photoreceptors because they do the detecting of the light, but they're very needy cells. And so the photoreceptors can't function or survive without their handmaidens, the RPE cells that are beneath them, which take away all the waste products that they make and also give them all of the nutrients that they need to function appropriately. And then those photoreceptors are involved in essentially like a 3-wire arc. So they connect to an intermediate cell type called bipolar cells, which then connect to a ganglion cell. And then the ganglion cell sends its long wires back to the brain, and then the brain does the seeing for us. So it's part of the brain, the retina is, and these are neural connections that are very, very delicate that allow us to see. So in Stargardt disease, in contrast, you start to see, as Dr. Commander mentioned, this kind of yellowish, these yellowish deposits. Hopefully you can appreciate that it doesn't quite look right? You have these little blackish areas too, but mostly you see this kind of yellowish region that's in that center, that's encompassing that very central portion of it, that's the most important part of your vision. And so let's go back to our schematic then, and what's going on in that area right there in the center, just to explain what we're seeing when we look into the eye. And to help with the schematic, I'm going to show like a visual field. So this is a nice fall scene from northern Wisconsin. So that's where I am here. So I thought I'd use it. So early, what you see in Stargardt disease is the accumulation of these yellowish deposits. They're like fatty deposits, and they occur kind of in the retina itself, but they're almost like little poison pills. So the photoreceptors are unable to clear out or handle these kind of tiny little poisonous molecules that normally, if we have our normal genetics, we're able to handle and get rid of. But in the case of Stargardt disease, these things build up. And it's the nature of photoreceptors to shed part of their distal segments, which are then taken up by the RPE cells. And so because they're the handmaidens, they clean up the garbage. And so the photoreceptors are making all this extra kind of nasty garbage. The RPE does its best to ingest that and try to break it down, but it can't break it down. And so what happens over time is that the RPE cells, those handmaiden cells, break down and die, which secondarily leads to the photoreceptors dying too, because they can't survive on their own. They need their RPE cells. And when that happens in the center of the vision, that's where the portion of your visual field starts to deteriorate as well. Now, once you're in this position, you no longer have RPE cells or photoreceptor cells even around. So drugs aren't going to work. Gene therapy isn't going to work, because those are designed to slow, halt, or make the cells, the photoreceptor cells or the RPE cells, do a better job. Well, if the cells are no longer around, there's nothing left for those interventions to act on. So in that case, you have two options. One is to bypass the whole system. So is there a way to get other cells that are still alive to detect light and maybe do part of the same job that the photoreceptors used to do? And one approach to this is called optogenetics. So we're going to talk about bypass first. One approach to this is optogenetics, and this uses a kind of modified bacterial protein, and it injects, similar to what Dr. Commander showed you, injects these particles. They get into either the ganglion cells or the bipolar cells, and they convert those cells into light responsive cells. So they can now respond to light when before they couldn't, because they allowed the photoreceptors to do it. And so this is one promising avenue of investigation that could restore some vision to patients with areas of vision loss. Another approach would be prosthetics. So these are actually kind of electronic chips. So you can either place them on the surface of the remaining retina or beneath the retina, and they can detect light either through glasses that people wear that then project the light into the eye to stimulate these electronic chips, or some of the newer ones themselves may even be able to operate quite nicely with ambient light that just enters the pupil like it normally would. So this is also another exciting way, and says, hey, we don't have photoreceptors, we don't have RPE anymore, let's try and deal with it. Now, the other way to approach it is to try to replace those cells. So if they're missing, is there a way for us to be able to replace them? And so this is where stem cells potentially can come. Now, before I go too much further, I want to kind of define stem cells a little bit. People throw that term around an awful lot, especially folks that are trying to sell you things at the corner stem cell clinic that seems to pop up everywhere. And they try to confuse people using this term, thinking it's some catch-all magic treatment for everything that ails you. And so there's different things that people call stem cells that can even be considered stem cells. Some of them are tissue restricted, which, so for instance, your skin, when it sloughs off, your skin can make more of itself. And so the skin naturally has stem cells in it, but those cells can't make anything but skin. Similarly, your blood renews itself over and over and over again throughout the course of your life. So in your bone marrow, there are stem cells that create those blood cells, but they can't make anything but blood. And then there are also sources of fetal cells that people will also use on occasion. And those can perhaps make different cell types. If it's fetal retina, they could make a small amount or limited amount of fetal cell retinal cells, but they can't be made in large amounts so that they can treat lots of individual patients and still retain their qualities of making cells as complicated as photoreceptors. So that leaves us with what are called pluripotent stem cells. And pluripotent stem cells really do have the capacity to make every cell type in the body. The question is, how do you harness them in a direction that can make the cell type that you're interested in? In our case, the photoreceptors in the retinal pigment epithelium. And that can be a real challenge. So I'm plugged for the University of Wisconsin again, too. I was very proud that Jamie Thompson is the person who first invented or discovered human pluripotent stem cells back in the late 90s. And he recently retired, but I learned an awful lot from him over the years. And I'm very grateful to be at UW because of the work that he started. There are two main types of pluripotent stem cells that derive from human. One is called an embryonic stem cell. That was the first type that came out in the late 90s. Those are derived from a mass of cells that are present in very, very early embryos that are frozen for in vitro fertilization. And they can be used to create embryonic stem cells. Then later on in the mid-2000s, there was an ability to take skin cells or blood cells or lots of different types of cells from any human on the planet and to use a way of genetically reprogramming them to become cells that are essentially identical to embryonic stem cells. And because these are pluripotent stem cells that were induced to become pluripotent, but originally were not, they're blood or skin, they're called induced pluripotent stem cells or iPS cells. So in that case, with iPS cells, we typically take a blood sample, like you might submit for a cholesterol check. We reprogram them genetically and then grow them in a dish. So these are cells that have never seen anything but the inside of a plastic laboratory dish. And then over time, they will produce these colonies of iPS cells. And they don't look like much. This is a single colony under a microscope of thousands and thousands of these individual little iPS cells. And they're just round, nondescript. But each one of them has the ability to make potentially every cell type in your body. And again, like I said before, that's an amazing ability, but it's also a very daunting task to be able to take these rogue cells and get them to do exactly what you want them to do in a dish. And many years ago, that was worked out for retinal pigment epithelium. So that support cell, that cell that dies first in Stargardt disease, it was amazing that we could make these cells, and they look like these little brownish, blackish stop signs. And so again, here are thousands of them, all kind of grouped together and tightly packed, just like a retinal pigment epithelium should look like. And not only do they look the part, but they also can function like a regular human retinal pigment epithelial cell monolayer. And so that was worked out relatively early in the 2000s, but making photoreceptors out of retinal tissues was took a lot more effort. And so work from a lot of labs, my own and others, showed that you could make these three-dimensional floating, they're about as big as a big pinhead, these little retinal tissues, and you can make thousands and thousands of them. And they are actually arranged just like a real human retina. Here's what they look like if you cut it in half and look for stains for specific cell types. So they can look really quite elegant. And over time, and this is a picture here of one of those retinal organoids, or those tiny human retinal tissues. And we start to see photoreceptors being made relatively early. These are the dark gray or black spots that you see here are actually newborn photoreceptors. And if we wait a little bit longer, they start to move towards the outer portion of these organoids. And they start to make more and more of them and pack in until you have what it looks like, again, that layer of photoreceptors that are on the surface. What we don't see, though, is the RPE. The RPE we grow separately. So unlike the real human retina, we don't grow them together. We grow them separately. Now, if we let these grow really long, so almost as long as it takes to form a full retina in utero, then we start to see that they're making the types of structures on the surface that are needed to detect light. And blowing that up really big, so this is each one of these kind of red alien-looking things is a single human cone grown in a laboratory, entirely in a dish. And so they have different parts. Now, how close does this look to the actual cones that are in your eye and my eye? Well, I don't think anybody's going to say that they're as pretty as real human cones, which is shown on the left here, which is a figure from Dr. Swenka at the University of Alicante, and those are real human cones. So they're certainly not going to be the belle of the ball, but at the same time, it has all the features. So it has an area that can be used to detect light. Another area below that produces energy. Remember, these are the most highly energetic cells in the entire body, so they better be able to make a lot of energy. They have an area called an axon that does the wiring. And then it has a terminal process where it makes connections with other neurons. So it has all of those structural areas, but do they actually function in a dish? And so we did a lot of work with a lot of collaborators here at UW-Madison to show that indeed, if you take a special electrode and you place it onto one of these cones, of which there are hundreds of thousands in one of these organoids, and this is Dr. Raunik Sinha's expertise here at UW-Madison. So he's able to poke one of these with what's called an electrode, and able to show that in response to particular wavelengths of light. So in this case, this is a cone or a photoreceptor that is designed to respond to a green light. And so if you give this cell red light, it says, I don't care. You give it blue light, it says, I don't care. You give it green, and bloop, it makes a nice signal. And if we compare that with the same types of cones that we have in our own eye, it's not exactly the same, and not all of them work, but a number of them work almost as well as the ones we have in our own eyes. So that's exciting, just to show proof of concept that these aren't just photoreceptors that look right, but actually can function right as well. Another example would be the wiring. Here's an example of one of these early cone photoreceptors with its wire looking out. There's nothing around it, so it doesn't have anything to connect to, but it certainly looks like it's searching for a purpose in life. So once we and other laboratories did work to show that these cells have at least all of the bells and whistles that could be useful in trying to replace something as complex as a photoreceptor cell or an RPE cell, which I'm biased, but I believe are some of the most complex cell types in the entire body. So the fact that you can grow these from something that doesn't know what it wants to be at all the way to something that's a specific wavelength, color, light detecting entity inside of a plastic dish is quite remarkable. But we have to then be able to take that technology and move it from the laboratory into a facility that allows us to ramp up production so we can make enough, billions and billions, to treat patients, and to do so in a safe manner that's okay to be used in humans. And so that's where we worked with a local company, and actually not a lot of companies now, to be able to ramp this up into bioreactors. And so what you're seeing here is a single 3-liter, like a little bigger than a 2-liter of soda pop, but it's filled with photoreceptors. So human photoreceptors derived from iPS cells. And these can be grown in tandem so that hundreds of billions can be made at one time and cryopreserved. And so that was a big step forward to being able to bring this towards clinics. What about RPE cells? Because I said in the beginning that it's not just one cell type that we need to replace in advanced disease, but perhaps two. Well, RPE cells have been in clinical trial now in humans for quite some time. There's upwards of, I think, about a dozen that are either ongoing or have been performed in humans, and they've been safe thus far when they've been in these clinical trials. And so we can make those as well, and we make them in large batches. So here's a giant IV bag full of human RPE cells. And so ramping this up and doing it in a clinically efficient manner and a manner in which it's suitable for human use has been done. How about getting this into patients? So it's one thing to make them and to have them in the freezer, but how are we going to actually get them into the back of the eye? And Dr. Commander showed you some videos of how to get in viruses, but cells and these tissues are much bigger than that. And so that causes a problem. There's potential for more damage when you put it into the subretinal space. And so we have to weigh that safety versus potential efficacy. One way to, and we also want it to get into a certain spot. We want it right there, right in the macula. So if we can help it, we want to deliver it to where it's needed and not to some other place that's actually functioning okay. And one way to do that is to employ a biomanufactured scaffold. So these are synthetic scaffolds that are compatible with the subretinal space. And that sounds easy to do, but actually it's very hard because, especially if you want them to dissolve over time, you have to make sure that it's all compatible with our very, very delicate retina.
Dr. Gam, you're at the 18-minute mark. You should take 2 minutes and wrap up.
Okay, sounds good. So what we did is we worked with another group here to engineer scaffolds, and we used these kind of honeycomb type scaffolds. And in these honeycomb scaffolds, which are really, really thin, about the size of a dime, we can line them with those retinal pigment epithelial cells. And this is what it looks like. Each one of these stop signs is a retinal pigment epithelial cell that's lining one of these wells on one of these scaffolds. And then on top of that, we can take some of the cone photoreceptor cells that we make in the laboratory or in industry and seed those into them as well. So hopefully we have something that is more conducive to delivery into the subretinal space. And then the next step is to test, and we work with a lot of groups to do this. This is a group led by Dr. Barty at the National Eye Institute, showing us putting one of these scaffolds into the subretinal space of a pig. So in summary then, what I want you to kind of walk away with is that in late stages of Stargardt, drugs and gene therapies, which have tremendous promise in the early and mid stages, kind of lose their potential benefits. And so we have to think about how to bypass or replace the cells that have been lost. Replacement of this requires both RPE cell replacement and photoreceptors, and in particular cone photoreceptors, because that's what populates that center of our macula. Clinical trials are underway for RPE, and they should be starting very soon for photoreceptor replacement, but they're doing them individually at first. And next step would be to be combining those two to help folks like the folks with Stargardt disease. And I also want you to know that you're not alone, and that a lot of times folks say, well, you know what, everyone gives all this attention to macular degeneration. And for those of you who are Brady Bunch fans, you can kind of remember this whole Marcia thing. Everyone says AMD gets all of the money and all of the attention, but really the same approach is agnostic towards whether you have AMD or Stargardt disease. So advancements in AMD and with regenerative medicine actually help Stargardt patients as well. And then just to finish up with the few seconds I have remaining, just some parting comments and cautions. One is, what types of cells are we going to use? People talk about using them from individuals. That's possible, but very, very expensive. So it's not really ready for prime time yet. Perhaps we can make one cell line that can be okay for everybody. That might require immunosuppression, so we have to think about that in terms of safety. Or maybe doing something in between. And also expectations. No one is going to be able to restore all of your vision, people's vision, with the first shot. This is technology that's going to take time to grow and get better over time. We hope to make meaningful improvements in people's lives, but it's going to take iterations and going back to the drawing board and making it better to make the technology reach its pinnacle. And then while we're doing that, please be aware that there are a lot of snake oil salesmen out there that are trying to sell you a bridge in Arizona, and be careful when somebody promises you something that sounds too good to be true. With that, I just want to thank just all the different folks that have been involved in all of this work, and thank you for your attention.
Thank you, Dr. And this was very insightful discussion. With that, I'm just going to open up for the questions, and I'm looking at it in the Q&A at the moment, and I'm not seeing any questions. So I'll start with my questions while we are waiting for the questions to pop up in the Q&A box. So since you're the last speaker, I'll start with you. What are right now the current challenges with the engraftment? Do we have any animal data on that with the engraftment of these transplanted stem cells and the longevity in terms of?
There is. So from an engrafted standpoint, in terms of the survival, delivery, extension of axons, interactions with host retina, that's been shown in mouse, rat, for our group with pig, non-human primate, and dog. And so in all those scenarios, what's interesting is that the cell types that are put in the subretinal space, regardless of the animal model that's used, the maturation program is human. So it follows the same length of time that human cells would normally require in terms of expression of opsins and other phototransduction proteins, and elaboration and elongation of outer segments. So there's quite a bit of data with regard to that. In animals, of course, these are xenografts, so they have to be immunosuppressed. In rat models, where you eliminate their ability to reject foreign cells, they survive for upwards of a year, as long as we've looked at them. In other animals where we don't have the benefit of having them genetically modified so that they can accept any cell type, they can survive as long as we can keep the animals happy and healthy. But then they have to be immunosuppressed.
Thank you so much. Because while you were presenting, and I was just thinking about the stem cells in the angiogenesis, and there were a lot of lessons learned from there. Are there any still like kind of a thought process or consideration for the possible side effects of the tumorigenesis, like these are the implanted cells and what path do they do or once they implant?
Great question. The answer is really no. There's been a lot of trials already with RPE cells derived from pluripotent stem cells. None of them have shown any sign of growth, unregulated growth of cells. Also, the FDA requires us to do extensive testing. And in order for us to even see a tumor, we often have to spike the product with upwards of 20 to 50% of undifferentiated pluripotent stem cells. It's actually very hard to get the products to develop a tumor, and that has to be rigorously tested before you can get into human clinical trial. So really the thought process between, or the original thoughts about, oh, these can cause teratomas or tumors have not shown to be correct, at least at the level of production sophistication that we have today.
So I have a few questions in the chat box. So I'm just going to address these. The first one is, are you investigating ways to reset the epigenetic state of donor cells to improve their integration and function in the host retina? I think some of that you have addressed already, but if there's some more you would like to.
So I mean, again, it's one of those things where you would address things that you need to address up front. So right now, we can make all the cell types that we need. And so usually when we're talking about epigenetic marks and things that can be barriers to cell production, it's preventing them from making the cell type that we want. I think the other thing that the person asking the question is getting at is, can you eliminate things that maybe make it look different than what the host is? So there's a complicated factor for that, because if you do that, epigenetic marks are important. They're important for determining what your cell type is, how you function. So if you eliminate them, you're going to, that would be eliminating them throughout the genome, and that would greatly affect the cell types themselves, whether or not we could even make them. And yeah, I'll say also that the FDA, the more you manipulate the cells too, the more it brings up ideas. Well, you might move it over here towards something that might be positive, but then what are all the negative things that you're doing at the same time? So all that has to be considered, especially when doing something that's a first in human.
Great, thank you. The second question is, I think this is for Dr. Commander. Is there a progressive element in Stargardt disease? Does visual acuity decreases in the 4th, 5th, or 6th decade?
Yeah, that's a good question. So my original teacher about this said that he noticed two patterns, that there's people who progress for a while and then level out, and people who continue to progress for their whole lives. I think with the modern imaging that we have now, we can see that everybody progresses very, very slowly, at all parts of their life. But I think the way that kind of the idea that some people level out for a while or level out is that it affects people in their lives in kind of like a stepwise way. So even though the tissue might be changing at a very specific rate inside the eye, the patient's perception of it and their reality of it is, well, I was fine until I was, 35, and then I had trouble doing X, and that's when it got really bad. And then it got really bad when I was 55, when I couldn't do Y, but it's been the same since then. And if you look, after age 55, the little spot, that is diseased in their eye, would expand just a little bit more, but maybe it hadn't affected their life activities more, so it can seem that it's topped out for them. But the short answer is, yeah, it does. The area that's affected does expand over the course of your life. So, I mean, all of that is, assuming no treatments available. So I'm hoping that by the time, as people are projecting out their own lives, however many decades, that we have better treatments for you guys.
Thank you. Thank you, Dr. Commander. Another question for Dr. Gam is that has the organoids plus RP combination been ever tested in animal models?
We're not leaning towards organoids and RPE because organoids make all the different cell types of the retina. And so we're trying to be a little more directed at just replacing the photoreceptors or introducing the photoreceptors in RPE. So it's really more of a purified photoreceptor precursor product. But yes, so those are our, the examples I showed you. We're doing that in the laboratory. But it is more complicated, as you might imagine. The more you add to the equation, the more different cell products, putting them together, adding a scaffold, all those things have to be made as economically as possible, as safely as possible. They all have to be tested. So adding different elements to any product is going to lengthen the runway in which it'll need to get to the clinic. But things are moving along.
Thank you so much. I think in the interest of the time, Brian, probably I thank you. Thank you for all of you for arranging this webinar. It is very informative, and I'm sure people who have attended it got the benefit of the additional new research which is ongoing in this therapeutic area. So Brian, back to you.
Thank you very much. And I just want to thank you, Dr. Khan, for your very able moderation, and our two speakers, Dr. Commander, for giving us a very good overview. And I love the live surgery videos and some very promising approaches there. And Dr. Gam, really exciting developments in terms of regenerative medicine and stem cell approaches. It really is almost like science fiction coming alive. So we wish the best for these promising treatment approaches going forward. And I want to thank everybody who participated in this presentation by serving as an attendee, by coming. We have recorded the session. We will have a link that will be sent out to all the participants next week. And of course, this is the Lunch and Learn event will be something that will go on our website, and there'll be many other people that will look at it as well. And I'm sure that Elkias Pharmaceuticals will send it to their audiences, and Dr. Gam and Dr. Commander will urge you also to help to disseminate it. But thank you all, and it's been a very informative and very promising session in terms of the treatment approaches that are being developed. So thank you, and we wish all of you the best in terms of coming up with effective treatments for this very, very serious and very debilitating vision condition. Thank you.